Hi everyone, welcome to the lecture on diffusion MRI for Enigma-U. So here's an outline of what I'm going to talk about today. I'll start with an introduction to what diffusion MRI is and talk a little bit of physics. We'll go through some of the common sources of error or noise that we need to control for, some of the advantages of diffusion MRI and drawbacks, and some of the common uses. So first, some terminology. As with many scientific fields, we've got tons of acronyms, and with diffusion MRI, this is no different. There are a lot of acronyms here, some of which are interchangeable, and some of them are refer to specific acquisitions. So the general terms are diffusion MRI or DMRI or diffusion weighted imaging, DWI. These can pretty much be used interchangeably. Some specific terms that you might occasionally see used in a general sense, but they are supposed to be specific, are diffusion tensor imaging, DTI, high angular resolution diffusion imaging, HARDI, diffusion kurtosis imaging, and diffusion spectrum imaging. And these ones refer to specific ways of modeling the data or specific resolutions of the data. Some of these have, as the name suggests, higher resolution to identify differences in white matter structure, or they model the diffusion signal a little differently, perhaps to be able to disentangle crossing fibers, which is a common issue with more basic DTI acquisitions. So first, what is diffusion MRI? Diffusion MRI can measure the direction and magnitude of water diffusion in the brain by measuring Brownian motion of water molecules. Brownian motion is just the random movement of small molecules in a fluid or a gas. So for example, in this image here, in an area where there are no boundaries, you're going to have water just moving randomly around the space. If there are barriers present, however, such as myelin surrounding the axons, or cell bodies or other barriers within the space, this will change the magnitude and or the direction of water diffusion. We model this diffusion as an ellipsoid. We do this for each voxel of the brain, and a voxel is our unit of measurement in the brain. A common voxel size for diffusion weighted imaging might be 2 millimeters. So if you imagine for every 2 millimeter cube space in the brain, you have an ellipsoid like this. We can model the direction of water movement in three primary directions. So here, for example, in a well-myelinated tract, we might have a fairly elongated ellipsoid because water is diffusing more easily in one direction rather than randomly across other directions. And with this, we can also look at just changes in the magnitude of diffusion, how much movement there is just in general. So just a little background or a refresher on neuroanatomy as we're primarily talking about white matter. This primarily concerns axons and myelin. The neuronal cell bodies are mostly in the gray matter. So recall that around the axons, we have these Schwann cells in the central nervous system that are cells that surround the axon and make up the myelin, the fatty insulating layer that helps axons transmit their signals. And in a brain, this is what it might look like. This is in a dissection. So if you imagine here on this uh, slice here, if you imagine cutting through this blue line, this is what you might see. And we call this area the corona radiata, which kind of looks like a crown radiating out. So uh, this is what we're measuring with diffusion MRI, essentially, what we're trying to recreate. So as I just mentioned, myelin insulates axons, and this preserves information flow. Just like in electrical wiring in a house, you insulate the wires, one, for safety, but two, so that information, the signal is able to transmit with minimal signal loss. The exact same thing goes on in the brain. And here, this is just an image of my brain actually, where all of the white matter is in blue, the gray matter is in gray, of course, but just to show how it really forms the core of the brain and it connects all of these different brain regions. So we're going to talk just briefly about the physics behind the diffusion MRI signal. Recall what happens to protons in a strong magnetic field, what we call the magnetic resonance effect. The most common source of protons in our brain is hydrogen, as they are part of water, and Water is everywhere in our brain. And so with these hydrogen protons, all of these protons are spinning. This is what they do, and they all have an axis around which they spin. And these are distributed randomly. So you'll have some spinning this direction, some spinning this direction, and so on. This is what occurs when there's no magnetic field, just um, the brain existing the way it is. But when these protons, i.e., the body, are put into an MRI scanner, these protons align to the main magnetic field. So instead of spinning in all different directions, they are now all spinning in around one axis, either one direction or the other. So what we do in MRI imaging is we can apply a radio frequency pulse, and this makes the protons flip in their orientation, and now they're spinning along a different orientation. Most of the signal that we generate to create, most of the signal that we collect to generate these images comes from what happens in between these pulses and when the pulses go off and things relax back. And so this is what we are taking advantage of when we do magnetic resonance imaging. So what's different about diffusion MRI? That was just basic MR physics. With diffusion MRI, we add a gradient magnetic field, meaning we add a magnetic field where the strength of the magnetic field is not constant across the area that's being scanned, so across the brain. We might have some parts where and this varies linearly, so you have weaker signal on one end and stronger signal on the other. The speed at which these protons precess or spin about their axis is based on the magnetic field strength. So when you have a stronger magnetic field, they're going to spin faster. If you have this varying magnetic field strength across the brain, you're going to have different spin speeds. We can say now that these protons are dephased. They are not spinning uniformly. You have some that are spinning slower and some that are spinning faster. We then apply another pulse to rephase these protons, trying to get them back to the same spin. But some of these protons, the ones that have been spinning the fastest, may have gotten so dephased that they don't fully rephase before the next dephasing pulse is given. And this results in signal loss. Now, signal loss here is not a bad thing. This is what we're taking advantage of to actually create these images. The signal loss depends on how much water is present in the tissue and how fast it is diffusing. So if you imagine in this image here, you have an area where it's primarily CSF, so you've got very fast diffusion in all directions. During the dephasing, you'll have some protons that get wildly out of phase, and you can see a big difference between those and the ones that have not dephased. In other areas, and so this leads to a lot of signal loss because they're not all able to rephase. In other areas of the brain where there's less water or there's less water moving along this direction that we're looking at right now, and I'll explain what I mean by that on the next slide, that you will have some who get dephased, but they don't get dephased that much. And so they're able to rephase during the rephasing field. And so we don't lose information along that direction. So this is all talking about one direction, where you're looking at this image, and basically the areas that are dark are the areas where we have lost signal. And that means that water is moving a lot along the direction that we're looking. But you can't do a whole lot with just one direction of information. If you imagine you are trying to calculate or count the number of cars at an intersection, how fast they're moving, how many there are, you're going to be able to see cars that are moving some directions and estimate how many there are and how fast they're moving much better than you are going to be able to estimate along other directions. So what we need is this information at multiple directions. And this is exactly what we do with diffusion MRI. If you imagine this sphere around the brain, and each of these points are points at which we essentially take a picture of the brain. So you take a picture from one direction, you take a picture from another direction, and with all of these, you are able to recreate this information about water movement because you have all different pieces of information about movement in different directions. So the way this looks in the brain is you might collect a single volume that looks like this, where areas that are bright are areas where we have not lost signal. So this would be areas where there's less movement in that direction. But then we have other ones like this, and so on down the line. And once we are able to combine these, then we can create a comprehensive picture of movement in the brain. And this is what this might look like after you've combined all the information. This is one of the types of images that we can get out of this. With these images, the color indicates the primary direction of diffusion. So red is tracts that are oriented left-right, and we see that most prominently here in the anterior and the posterior parts of the corpus callosum, the genu and the splenium, which makes sense because the corpus callosum is the main conduit between the left and the right hemisphere. So of course, these are going to be a lot of left-right running tracts. Green is anterior posterior, so tracts that run from the front of the brain to the back of the brain. And again, these make sense with these tracts that go along into the occipital lobe. And blue is superior inferior, or things that run from the top down to the bottom. So the ones that we see most prominently here are the internal capsule, which is part of the which leads into the corticospinal tract. So these are the tracts that run kind of as we saw with the corona radiata, that come down through the internal capsule, down into the corticospinal tract and out to the rest of the body. So what are some common sources of error or noise that we have to try to either mitigate or account for, or that in some cases, unfortunately, mean we have to remove data? Noise, as it sounds like, this is noise in the image, just like you can have noise in any signal. This is inhomogeneities in the signal, and this is what it might look like. You have this just kind of fuzziness around the brain. We can fix this with a process appropriately called denoising. So this is an example of what an image might look like after denoising. Another one that we need to account for is called eddy current distortion. And what this is essentially, it shows up as movement between the volumes because we collect all of these volumes at different times over a 5 or 10-minute scan, and there's going to be some movement in between these. So what eddy correction does is it aligns all of the volumes to the first volume. Here on the left, we see a volume that has not been eddy corrected, and on the right, we see one that has. So you can see the kind of jitter in between the images, and this has been mostly fixed in the other one. So this means that we are better able to model the movement of water since we are having to model the movement of the slice itself. Motion artifacts are a common source of errors. Motion can lead to poor tissue differentiation, as you can see here in this one. It's very blurry. You really can't tell a whole lot. And or they can lead to signal dropout. So this is one example here. Signal dropout, well, it might show up in this coronal slice as a black line across the slice. And if you look at that axial slice, you can see a lot of the image is very dark. And this usually happens because someone has moved and we haven't been able to collect the information that we need. Now, some of this it's pretty common to see this, and it's not always a huge problem. You know, if it's just a few slices or a few volumes, usually that's okay. There can be some correction possible to interpolate or to try to recreate missing information, but sometimes it is necessary to drop the data, unfortunately, if it happens in too many slices and too many volumes. So this is why limiting motion during the scan really is critical. Because if we just haven't been able to collect the information, there's sometimes limited options to approximate it or recreate it. Epi-induced susceptibility. So what this looks like is this kind of blurring or stretching in one direction across one axis in the brain. It is most often seen at the air tissue interfaces, so the prefrontal cortex, which is right above the frontal sinuses. This is often where we see this sort of distortion. And here you can see it clearly in these images where some have, you can see the stretching forward and in this case, kind of squishing inward. So there are two ways that we can correct this, and often we do these two things together anyways, but registering the image to an anatomical image will help kind of squish things back into the way that they're supposed to look. But the preferred way, is to collect field maps with opposite phase encoding. And what we mean by this is that if we can't do away with this distortion in the signal, and we know that it's happening along one direction, we can collect it across the other direction, and then we can approximate the difference in the two and use that to try to correct the image. So here we have two field maps where, as you can tell, the sort of distortion that they have is opposing. One of them is stretched forward, one of them is squished inward. And when we use the information from these two, we can correct the main diffusion volume. So here's what this looks like. As I said before, we have this distortion primarily in the prefrontal cortex, and afterwards it's been largely fixed. Some of the advantages of diffusion MRI, like other MR sequences, it's non-invasive. It can be collected on the standard MR scanner. You don't need special equipment or special software for the most part. It's using, needs just tweaking some parameters. There's no contrast agent necessary or no chemical tracer, so there's no injection. There's still no radiation exposure, as there is in all MR scanning, no radiation exposure. It does not require a participant to do a task or even to remain awake. So this can be really useful for young participants, for participants who have trouble staying still. This can be useful in a number of clinical populations. And it is more sensitive to microstructural changes than standard MRI, by which I mean standard anatomical imaging like T1 weighted imaging. This is particularly useful in disorders where we expect to see microstructural changes. So I am a TBI researcher. We know that the white matter is particularly susceptible to damage after a brain injury, where when the brain experiences a force, these axons are stretched and this disrupts their integrity. Sometimes they can even be broken, but that's happening on such a small level that you usually can't see it on CT scans or on T1 weighted imaging. You can, however, see that on diffusion weighted imaging. So there's been, in the last 10, 15 years, a huge increase in the number of papers using diffusion weighted imaging in TBI. Just as an example, it's been used in many other situations as well. Some drawbacks of diffusion MRI, it is scanner dependent. The numbers that you calculate from it are scanner dependent. So this makes it difficult to have comparisons across scanners or to establish objective thresholds. So you can't scan a participant and say, oh, this metric in the corpus callosum is 0.2, that's bad, because it really depends on the scanner. So you really need to have control, kind of healthy control information to be able to figure out what you might expect to see. But there are a lot of efforts underway right now to come up with new ways to harmonize data across scanners to enable these direct comparisons. As we've talked about, it is motion sensitive, so you have to do what you can to mitigate motion, but if there's too much, we just have to get rid of data, unfortunately. Any change in the acquisition parameters will alter the calculation of the diffusion metrics. This we'll talk about the specific diffusion metrics on one of the next slides, but we do need to keep the parameters consistent if we want to directly compare the measurements that we get. We have to also keep in mind that this is not a direct measure of myelin. We are mathematically modeling the white matter. This is the case in a lot of MR imaging that these are not direct measures. We are taking advantage of the signal and we're calculating things. And in many cases, they are very good approximations of those, but we just need to keep that in the back of our mind at all times, because it's useful to think of alternative explanations or, try to think about what's the neuropathology that you think is going on and how might that affect these measurements. And one less of a drawback and more of just something you need to keep in mind. The measures that you calculate from diffusion MRI are very age dependent. We know that white matter is continuing mature well into the 30s. And these changes are nonlinear as well. You have different periods where there's faster maturation or slower maturation. So really, you just need to keep that in mind and either have age appropriate controls. You need to include age in your model frequently, just something to keep in mind. So some of the uses of diffusion MRI. Going back to this image that we looked at on the one of the early pages, we can use diffusion MRI to assess the level of white matter organization. There are four basic metrics that I'm going to talk about and three more advanced metrics. The four basic metrics are fractional anisotropy, or FA, as shown here in this figure. This is the degree to which water diffuses along the axons rather than across it. So you're going to see high FA in areas where we have very intact, well-myelinated axons. So again, the corpus callosum is one of the areas of highest FA in the brain because we have this very strong coherence of left-right connectivity. Mean diffusivity, or MD, is the magnitude of diffusion. So going back to these ellipsoids, if you're measuring the diffusion in all three directions, mean diffusivity is just a magnitude, the average of these three. Axial diffusivity, or AD, is diffusion along the primary axis. Radial diffusivity is diffusion perpendicular to the axon, so the average of those two. And I just realized one issue here. These are switched. L1 is always the primary axis of diffusion. So this should say L1 and this one should be L2 and so on for these here. So the way these are usually associated, in areas where you see high FA, you typically see low MD, low RD, and sometimes high AD. FA is a ratio of the primary diffusion directions. In areas where you see low FA, you might see high MD and high RD. This might be in areas of the ventricles in particular, and in some areas, if you have restricted diffusion, but isotropically, meaning it's not primarily restricting it in one direction, you could have low FA, MD, and RD altogether. Some of the more advanced measures that we can calculate are ICVF, or intracellular volume fraction. This is a measure of neurite density. ODI, orientation dispersion index, which is how uniform the directions are. So this is kind of flipped from FA in some ways, where here you can see in the corpus callosum, the ODI is very low because things are primarily moving, again, in one direction. They're not dispersed in many directions. And ISO, isotropic volume fraction, is also sometimes examined. This is the free water diffusion. Just an example application. I already talked about TBI, but this is an example from schizophrenia, where when you compare individuals with schizophrenia to those without, there are a number of studies that have shown that there are widespread areas of lower FA in participants who have schizophrenia. You can see this here across the white matter. All of these orange areas are where this paper found lower FA in the schizophrenia group. This suggests that there might be myelin loss in schizophrenia or some loss of coherence of the fiber bundles. So one of the other things that we can do beyond calculating the different measures of white matter organization is we can model the white matter tracts. So going back to that image, similar to that image that I showed with the different colors, we can have an image like this that shows the primary direction of diffusion in these colors. But what we see for each of these voxels here, and a voxel in the brain is like a pixel in a picture. For each of these voxels, we can have these little lines that show the primary direction of diffusion. So here we're looking through the corpus callosum. And as I've mentioned multiple times, it's very coherent. There's not a whole lot of variation. We have fibers that are moving primarily left-right with a little bit of superior leaning here as they come up into the corona radiata. So we use this information to calculate FA and the other measures that I talked about, but you can also essentially play connect the dots when you have all of these lines here and connect them to make tracts like this. This is a tractogram of the brain looking just like this. So you can see the corpus callosum across the middle. We have the corticospinal tract going down. We have the tracts going into the cerebellum here. You can see the cingulate gyrus coming across the top of the corpus callosum. And in one of the following lectures, we will talk more about some of these, how to reconstruct these tracts and identify some of the tracts and their primary functions.